So as I told you, so there are other experiments also what we need to discuss. One is importance of chlorophyll. So chlorophyll, actually, in order to discuss the importance of chlorophyll in photosynthesis, what we have to do, you take two potted plants. One of the potted plant will have normal leaves. Other potted plant will have leaves which are variegated. What it is? It is variegated. The leaves are variegated. So what do you mean by variegated leaf? When a leaf is not fully green in color, but it is having white dots or some other white coloration, it will be called as variegated leaf. So I will show you how the variegated leaf looks like. So this is the variegated leaf. Hope you can see all the white portions that are there would be the variegated portions. So like this, what we are doing, you take a plant with variegated leaf and you will take a plant which is having normal leaves. So these two plants, we will keep it in sunlight for 2 to 4 hours, and then after that we do the starch test taking the leaves of both the plants. Then what is the result is, we find the leaf which is having complete green color will turn blue black after performing your starch test. Whereas the other plant which is having variegated leaves, in that only the green portions will turn blue black. As you can see, only these portions have turned blue black in color, whereas the other portions that are white in color will not develop any color. They will be negative for the blue color. They will be negative for the blue color. So this indicates that chlorophyll is needed for photosynthesis. If there is no chlorophyll, those regions will not show positive to the starch test. So which indicates the importance of chlorophyll. The next experiment what we have is related to release of oxygen. Whether oxygen is really getting released during photosynthesis or not, we are able to check it through the experiment. So for this experiment, we take water plants. It can be hydrilla plant or elodea plant. Okay, small plants with very small leaves you might have found in water. So such type of a plant is called hydrilla plant. So you take a twig of hydrilla plant, and what we do is we take a beaker with water and then you take a funnel. I hope you know what is a funnel. So funnel we will be used for filtering, using it for filtering and all. So that funnel you take, inside the funnel, okay, you keep the twig of hydrilla, whatever you have taken. As you can see here, we can place the twig inside, and then we will keep that funnel inverted into the beaker of water. So you will keep that inverted in this beaker of water. Okay, and then on the top of it you take a test tube. I have shown the test tube how it looks like. So you take the test tube, and what you do is you take water in that. So we take a test tube and we take water in that, and then you invert this test tube onto the stem of the funnel. That is, you have to, okay, invert that. You will put it, okay, from the top onto the stem of the funnel. You will take water in the test tube, and because of that water pressure, when you are keeping it on the stem of the funnel, the water all will be retained like that only. Okay, so this is the setup what we will do. We will keep this setup in the sunlight, and after 2 to 4 hours, what will happen is the interesting point here. What will happen after a period of time, children, you can see water is up to the brim in the test tube here. Okay, here what will happen after you keep okay, your setup in the sunlight after photosynthesis, you can see that the water has come down little bit, and here an empty space will be created in the test tube, wherein some air bubbles, okay, you can see coming out from the funnel. All that okay, you can see as a clear space actually in the test tube would be created. Your water would be pushed down. So why it is pushed down is because of release of oxygen. So how you can confirm whether it is oxygen or some other thing that has been released? So you know there is a simple test for oxygen, okay, release. So in order to check the release of oxygen, what you can do is you carefully lift the test tube and then put your thumb finger on that without the, okay, disturbing it. You should not allow the gas to escape. Then what you will do, you take a matchstick, okay, which is burning, just burn the matchstick and then take that and then remove your thumb finger and put it inside the test tube. What will happen? It will burn more vigorously. You know that when oxygen is present, the burning will be more vigorous. So it will burn more vigorously, which indicates presence of oxygen, and release of oxygen will be thus confirmed during the photosynthesis. Hope you understood. So you take a water plant, put it in a funnel, okay, because it will float away. We are keeping it in the funnel. On the top of that, you keep a test tube which is having water in it, and then put the setup in the sunlight. After a period of time, we find some empty space on the top. That is because of oxygen, and we performed them, okay, test to confirm the release of oxygen. So like this, children, we have, okay, all the experiments which are related to photosynthesis. One point you should remember is, whenever you are doing any experiment, okay, in that experiment we are not giving the plant only one particular component, whereas all other three raw materials are available for the plant. So that is an indication already, which that is the fact what you should already register. And one more thing, this is a very, very, very important point, and number of times it has been asked in the bits. Why? Okay, what we have to do before performing an experiment in photosynthesis is a question. So always, whatever plant you are selecting for photosynthesis, we have to keep it in shade. Where? In the shade for 7 days. Why you keep it like that? For 1 week or 7 days you keep it, because to ensure that all the previously made starch is all utilized by the plant, and plant is not containing any starch now. Because once we perform the experiment of photosynthesis, then only it should make starch, and then only our test will be confirmed, okay, for photosynthesis. Okay, so that is a very important point, that you should keep the plant in shade. Keep the plant in shade before the start of any of the experiments. That is a must thing what we have to do. Why that is done is to ensure that the plant is free from any stored starch before it's the start of the experiment. So this is a compulsory fact, and this is a very important bit that will be usually asked. So hope you followed all the experiments. A quick brush up of all the experiments you can do like this. So always take a pen and a paper and maintain a book for all the videos, so that you can have the continuity of what is being taught in the previous class. You can correlate, and when you are practicing also, you can just practice in that way. And when you are writing in your exam, use the sentence form and construct the entire answer. So in that way, you can learn the subject easily. So hope you followed, children. This is all very easy as such. Next, we will be going up to the next session. We'll have about the heterotrophs. So this is all about the autotrophic nutrition. So hope you followed, and it is quite easy as such, right? Okay, thank you.